Hi everyone, I'm Alex. I've been designing proteins using machine learning for about a decade now. Over the past several years, we've seen some really new, exciting protein generative models based on the transformer architecture. And in this series of videos, we're going to build one of these protein transformer models together from scratch. So in particular, over 5 lectures, here's what we're going to do. So for the first lecture, we're going to talk about the various aspects that you might need to consider when you frame this problem. And we're going to pick a data set that we can model using this approach. In lecture 2, we're going to build a simple protein language model that can generate new proteins. In lecture 3, we're going to expand our model to the transformer architecture. Then in lecture 4, we're going to learn how to build evals for our model to tell if they're actually any good for the things that we want to use them for downstream. And then in lecture 5, we're going to scale our protein transformer model using computational tricks, GPU compute, and we're going to be able to model all known protein diversity in the UniProt dataset. And so before I start off here, I want to give a huge shout out to Andre Karpathy for his Neural Network Zero to Hero series. That series was a huge inspiration for this series, both in sort of teaching style, obviously, as well as shirt selection. And in particular, we're going to go through a lot of the same steps that you go through in that series, even though our underlying data is quite different. It's protein sequences instead of natural language, but the modeling approaches are substantially similar. So huge thanks and shout out to Andre for that amazing series. Thank you. Okay, so our goal here is to build a generative model of protein sequences. And so for that, we're going to need some protein sequences to model. So for the purposes of this tutorial, it's too big a problem to model all known protein diversity. And so we're going to pick a specific class of proteins that are all related to each other. Similar to the way in the neural network series, we picked a data set of names that are all sort of related to each other in some way, and they're all kind of short and easy to model. We're going to kind of do the same thing here. So I thought a lot about what protein class to model for this series of lectures. Here's the one I kind of picked. So this is acyl phosphatase from Drosophila, and this is a crystal structure of it with the PDB code 1URR. So why did I pick this particular class of proteins for us to model? This class of proteins is relatively short, so it's just about 100 amino acids or so. And that's going to be good for us to keep things simple and keep things computationally efficient for the first couple lectures. Second, this is an enzyme. Enzymes are incredibly cool, the coolest proteins. And they also have specific residue features like catalytic residues and that we can use to do detailed evaluations of our model downstream. Also, this enzyme class has some functional data that's soon to be released. I saw a fantastic talk by Polly Fordyce from Stanford at NeurIPS 2023, where she discussed her lab's work doing deep mutational scans of this protein. And so hopefully, if we can model this protein class and create some useful models, our models will be able to predict the functional effects and mutations that will be released when her lab publishes that work. So those are some of the considerations that I thought about when choosing this protein class. So let's see if there's enough data in this class for us to efficiently model. And so to do that, let's turn to the most amazing protein resource on the planet, which is UniProt. So UniProt is incredibly densely indexed and cross-indexed, and we can just type in a PDB code here and immediately come to the UniProt entry for this protein. UniProt entries are incredibly detailed, and they're linked to all these other databases. So you can see in the entry for this protein here, the sequence length, the level of evidence we have for the sequence being real, the source organism, but also some really fantastic linking to the RHEA database where it shows the catalytic scheme here, the reaction this enzyme catalyzes, along with even kinetic parameters for specific substrates and things. UniProt is amazing, and we're going to sort of use it to collect homologs as well. So one of the things that each sequence is annotated with in UniProt is typically some kind of HMM-based annotation for like its protein family. And there's a lot of different projects that have sought to do this. And with two clicks here, we can family and domains and then acyl phosphatase family. We can get a big list of all the proteins in UniProt that are annotated to have this function. And this is going to be a great basis for us to start modeling. So there's 31,000, 32,000 examples here. I'm going to do a little bit of filtering here just in the UniProt interface. You can see that I have some additional columns in my view. I have the PDB cross-reference, so this will show you if any entry has a crystal structure. Fantastic information for any protein design campaign. And also, I have the active site annotations enabled here. This protein has sort of two catalytic residues, and they are annotated. Their position is annotated in many of the proteins in this database automatically using HMMs. This is really going to be really rich, useful information for us to build evals out of. So I would like to use it. So here's what I'll do. Let's restrict this list down to proteins that have this annotation, this active site. And because we're going to do a length filter later, we can just restrict this to sequences less than 200 residues. So this is sort of our raw data set here. 28,000 and change acyl phosphatase enzymes. They're from all these different organisms, so mammals, pig, rabbit, human, turkey. Turkey isn't a mammal. Cow, fruit fly, all sorts of bacteria. There are all sorts of tools in UniProt. We can examine this taxonomic distribution a little bit. But for our purposes, we'd just like a large data set of these homologous sequences, and we can get that just by downloading this to a FASTA file. So I've previously done this, and I saved this FASTA file here locally on disk, so I don't have to show what's in my downloads folder, because it's messy. So we'll pop over into the Jupyter Notebook here and dive into our data set. So first things first, we'll do our imports. I should say, just for computational setup, I'm using VS Code here. I'm running in just a totally vanilla Python virtual environment with Python 3.11. And I just installed these three packages: Biotype, read and write biological sequence data, Plotly for plotting, and then NB format is actually just so VS Code can run these notebooks for us. There are obviously a lot of different plotting packages for Python, and there are also a lot of different biological sequence reading and writing packages for Python. Over the past 10 years of designing proteins with Python, there've been I've noticed a lot of changes in tooling. These are two of the most ergonomic tools for me, so that's what we'll be using for these lectures, but there are obviously other tools that do the same thing. So let's import these, and then let's just take a look at our input sequences. So again, just recording what we did. We looked at the acyl phosphatase annotation, then we filtered those down to those with only active site annotations. We filtered those down to those that only have have a length of less than 200, and then we export to FASTA. And so this will just show us the first couple sequences in the file. We can see that the FASTA file is in standard UniProt format. So the headers have the database, the session, the entry name, some generated metadata, and then the organism and some other stuff that comes from the experimental data. when they collect the sequence. And the first thing I want to do is take a look at the length distribution of our sequences. And so to do that, let's go through the file and generate 3 new lists. So we'll have a list for the headers, a list for the sequences, and then a list for the sequence lengths. And then that list of lengths we can plot as a histogram. So running this code just loops through all the sequences in the file, and looks like we have 28,856 sequences in our raw file. Here are a couple of them. And then we can use Plotly to examine the length distribution. So this is the distribution of sequence lengths in our data set. It looks like the vast majority of our data is right here, around 90. And I think for the purposes of this tutorial, we can exclude most of the things outside that range. Also, just because we're working with sort of raw sequences from the database here, I'd like to apply a couple of other filters. I'll show you what those are here. So sort of the easiest way to do this is create a new empty FASTA file. And then loop through the existing sequences that we have, and then apply these conditionals. So first conditional is the sequence has to be within 64 and 128 residues. Second is it has to start with methionine. And third, we're going to exclude sequences that have ambiguous characters, which are represented in UniProt as an X. And we're going to write those sequences to a new file called acp.fasta. And running this code eliminates maybe 2,000 or so sequences, maybe a little bit more. So that's a hair less than 10% of our data set. I think that's a fantastic result to end up with 26,779 sequences to model. So this forms sort of the basis of our modeling problem. Here's the data set that we want to actually model. So just to recap what we did in this video, we went to UniProt, we got a list of homologs of an enzyme class that we care about. We downloaded those to FASTA. We looked through the FASTA at all the different sequence lengths. We plotted that, and then we made some choices about which samples to include in our data set. And then finally, we wrote that data set to disk for use in the next video. So in the next video, we're going to take this data set and we're going to build a very simple protein language model based on a paper from 2003 that can generate new proteins that look like those in our data set. So we'll see you in the next video.